Thank you very much. We definitely can take a quick question. And could I ask David and Fiona just to come and take a seat, and we'll convene the panel as well at the same time. Maybe just while we're convening, could I take moderator's prerogative and ask a question of the panel? With bariatric surgery, it's obvious that we can mount a suitable endogenous response. So why doesn't that happen in people who are gaining weight? Why don't we produce enough of these agonists ourselves? What's preventing that from happening?
It's what happens when you have bariatric surgery, as I showed, is you need that reorganisation of the anatomy of the gut to get the digestion and absorption of your meals lower down the gut, because that's the only way you can stimulate the GLP-1. So if you massively overeat, then you're probably going to get the signal, but that's not what you need to do, obviously, in obesity. So possibly some things you can do to the diet if you take foods that take longer to digest and that actually are digested lower down the GI tract, that will help. But nothing you can do gets these massive changes in the GLP-1 PYY that you get after bariatric surgery. I don't know any other condition where you see that. And you need those really high levels.
Yeah.
That's the key.
Thank you. Question here.
So Fiona, I have a scientific question. Is it known what is it about food absorption that triggers the release of GLP-1 in the enterocyte?
It's an endocrine cell.
Right.
So they're endocrine cells that make about 1% of the enterocyte. And yes, that's what we've been working on for 20 years, and I can talk to you about it for about 3 hours. But two simple things. You need the absorption. For glucose, you need the absorption on the sodium-coupled glucose transporter, and that makes a change. It depolarizes the cells, and they fire action potential, a bit like a neuron. And the rest of it is specific G-protein coupled receptors that detect fatty acids, amino acids, bile acids.
So there's a nutrient sensing mechanism
Yeah.
in that enterochromatin cell.
In the endocrine cells, yes.
And it's all charged up and ready to go. You don't
Yeah.
have to transcribe.
No. So they're very much like pancreatic B cells. They sit there with ready releasable granules. We need to increase the calcium, which you can do by these different mechanisms, and then they release. So it's an acute detect the nutrient, release into the bloodstream.
Thank you. Lady back here, and then John. Yeah.
On the subject, just to get a question to Fiona's basic science, on the subject of indirect effects, mitochondrial function is switched off by high glucose levels in tissue culture. Is it possible that by lowering the blood glucose, all the mitochondria everywhere start working better?
I'm not a mitochondrial biologist, so there's probably other people in the audience here who... I'm sorry, I really don't want to comment on something that's outside my field. Do you guys know?
I don't know the answer to that question, but I do know there's some evidence, or some people who think that GLP-1 itself can drive changes to mitochondrial function, but I guess that doesn't answer the question that you were asking. It's relevant.
You're sitting next to one. Yeah, I mean, GLP-1 receptor engagement does increase mitochondrial potential, so you do get an increase in ATP, ADP, but that's only where the receptor is expressed, and we've only really looked at it in beta cells, which are a unusual system because of their glucose transporter and glucokinase expression.
Thank you. I have a question, gentlemen.
So if you turn it on its head, do GLP-1 antagonists help people to gain weight and say they've got esophageal disease? And does that make Parkinson's and Alzheimer's and all the things that seem to do well with GLP-1 agonists worse?
I'm just going to repeat that question so everybody heard it. It was GLP-1 antagonists. Do they do the reverse and help gain weight?
So the only place I know where they're developing GLP-1 receptor antagonists is for people who've had bariatric surgery, who then actually, if you lose too much weight and your insulin secretion gets really bad, people go in the opposite direction. They get hypoglycemia after eating. And this can be so bad that people either say, please reverse my bypass surgery, which in some cases not possible. Some people end up having tube feeding because it's so bad. But there's a company in the United States that is developing a long-acting receptor antagonist for GLP-1 receptors, and that takes off some of these side effects. We don't know long-term what effects these are having, because in order to see effects on Parkinson's and body weight, you'd actually have to do long-term clinical studies.
Would it help their muscles recover? People who've lost muscle bulk because of the GLP-1 agonism?
I'm not sure that I would. It's an interesting idea. I haven't heard any companies who are thinking along that line, and the danger is that antagonising the GLP-1 receptor is probably going to reduce your insulin secretion, and probably will, I don't think it might not make you gain that much weight, because there are lots of other things controlling sort of weight loss, especially when you start getting into sort of multimorbidity and things. So, but I haven't heard any company that's thinking of it for anything except these massive side effects after bariatric surgery.
Could I just...
I'm going to set the down quickly for the Alzheimer's one. So, antagonist is obviously not widely prescribed. It's actually also in phase 2 for congenital hyperinsulinemia, because you just get turnover of the receptor, which is a similar problem, I guess, to bariatric surgery. But if we look at patients with variants in the GLP-1 receptor, which can be, I think, with increased and decreased function, I'm not aware that there's any signals in any of those patients of increased risk of Alzheimer's disease, so it's quite a complex interaction. And I think you've done the work in the variants, right?
Yeah, we have done some work looking through biobank type resources at the phenotypic effects of GLP-1 receptor missense variants, and the effects are generally pretty modest, actually, even on blood glucose levels and body weight. And I'm not aware of any conclusive data showing that there's any link to neurodegenerative diseases per se.
But bear in mind, if you knock out the GLP-1 receptor in mice, they're perfectly normal. Nothing is changed, glucose tolerance is fine, and it's because this is a very redundant system, so GIP receptor will upregulate, and I assume with patients on extended 9 you would see similar effects in the GIP receptor and probably even glucose receptor domain, so it's complex.
Just could I follow quickly with about the muscle loss and the other catabolic effects of these treatments, and how much of a concern that is, and do we think that, as was alluded to, that perhaps some of these combinations, and perhaps the partial agonists as well, might mitigate some of the muscle loss?
So I think it's a huge area of controversy at the moment. It's a hotly debated topic at the moment. Certainly when patients are treated with GLP-1 receptor agonists, some of these dual and triple agonists, they do lose a lot of weight, and a significant amount of that is lean mass. Not all of the lean mass will be muscle, some of it will be, fluid and well, maybe not fluid, but anyway, it's not all muscle. But I think, some people think that that's just an adaptive change, and there's some evidence that actually the quality of the muscle is improved despite the fact that there's less of it overall. To me, it would be maybe in certain groups of patients where you're worried about sarcopenia, maybe more the elderly and that sort of thing, this would be more of a concern. I'd like to see some actual hard data that shows that this is genuinely leading to increases in falls or decreased functionality or that sort of thing. I haven't seen any, but I don't know if you guys have or you're aware of any. It's definitely something that's hotly debated in the field at the moment.
Thank you. Well, I guess we're at the Longevity Forum. We know that lean muscle mass obviously is quite a strong predictor of health in an advancing age. But as you say, it doesn't seem there's much good data about the actual functional effects of that change. Thank you. Linda, do you have a question?
I had a question, but I've also got a roving mic, so I don't want to hug the microphone. But I did have a question about the ageing stuff. So in mice, when you're looking at receptor localization, presumably you're using youngish mice. Do you know what happens in old animals to the receptor localization, the receptor density, and also in people do different people have different receptor densities? Because some people have pancreatitis as a consequence of taking this stuff. Is it simply that they've got bucket loads of receptors and they're over-responding?
So I'll start with the last bit. So there is no signal for pancreatitis, and in fact that was a set of very controversial studies by Peter Butler's lab in UCLA, which didn't transpire in longitudinal follow-up. So anything you hear on the news related to pancreatitis won't be causal because there is no signal for that. So going back to the receptor levels, yes, mouse work is difficult in the UK. If you look at a mouse slightly funny, then the Home Office inspector will be knocking on your door. But we courtesy of our German colleagues, where they're not allowed to humanely cull mice until they're aged, we now have access to 81-week-old mice, which are incredibly old. And actually what we see is receptor expression is regulated in those mice, but the receptors are not functioning appropriately. Also in humans, and this is slightly contentious as well, we actually don't see much receptor at the protein level in human islets, for example. And I think that's because we're taking an islet from somebody who's born with the same beta cell that we're going to die with. And we think it's probably working on very reduced receptor levels because it's a really highly amplified system. And there's about to be some data come out which shows that maybe some of the effects are not at all on insulin for some of the drugs, and maybe more on appetite regulation. I think this just shows the importance of doing these kind of studies and careful, meticulous knockout studies like Fiona and Tina Muller's lab and others do to try and piece all this together, but also show the importance of using humans as well as part of this, because mice can't answer everything, and particularly the dual agonists, they're not the best model.
Yes.
It's okay, I can go, it's fine.
No, it's...
Oh. So, on the topic of the...
Is it on? Is it green?
You should have a green light.
Hello? No.
Oh.
On the topic of... There you go. The GLP-1 knockout mice. The GLP-1 knockout mice, when you give those guys the therapies, the GLP-1 agonists, do you see, so for the presumed off-target effect or things like that, how does that affect the sort of distal organ systems without these receptors?
So effects of GLP-1 agonists in the knockout mice.
The GLP-1 receptor is, so they're not really off-target effects, they're on-target effects you don't want. So they're not independent of the GLP-1 receptor, they're just side effects that are caused by GLP-1 receptors in places where you really don't want the effect. So for semaglutide, I'm actually not aware that it has off-target receptor effects that are independent of the GLP-1 receptor. It's the problematic ones are the side effects that are due to the GLP-1 receptor, but you target all the GLP-1 receptors wherever they're accessible when you use the drugs.
Thank you. Was there another question at the back yet, or if not? I have one as the microphone's going over quickly. Do these peptide hormones potentially converge on common downstream targets that could become useful drug targets, particularly for small molecules with maybe better sort of absorption and penetration to CNS? Is that?
I think all the ones we talked about today are GLP-1 and the other receptor targets. They're G protein coupled receptors, GS specifically, although not exclusively. So they all converge on similar intracellular signaling cascades. I'm personally not aware of anyone who is sort of trying to target those rather broad, groups of kinases which are activated by these particular G proteins, but I guess it could be an interesting thing to do. I mean, because they're so ubiquitous, you want to have some way of targeting them in a very cell-specific way. Some people have used GLP-1 as a way to sort of hijack its relative tissue specificity to deliver other therapeutic entities to specific groups of cells. For example, into the hypothalamus in the brain, you can use it to deliver corticosteroids, which can reduce brain inflammation supposedly. Similar things, similar process being used to target other organs, for example, using the glucagon receptor.
Thank you very much. Question at the back, and then one more at the front here.
Hello. In geriatrics, there's a need to reduce the amount of medication we are using to reduce iatrogeny or reduce the cost overall. Are there data that show a combination of, let's say, for example, physical activity and the usage of this drug that could suggest that we could reduce, for example, the amount of injection of or the concentration of these drugs?
So I didn't quite catch the question.
You said you could lower the dose of drug if you put physical activity.
Combined with exercise,
Right, yeah,
to lower the
synergistic.
dose.
I think generally at least for the obese population, generally people are wanting to see more weight loss than you can even achieve with, at least with semaglutide. So if the drugs are tolerated, then people tend to try to increase to the higher doses. I think that there's a strong feeling that exercise is an important component of what people should be doing if they're taking these drugs, partly because of the concerns about sarcopenia and lean muscle loss, and also maybe a way to try and preserve weight that has been lost over time. If people then subsequently are not able to keep taking the drug, either because in the UK they're only approved for use over 2 years, or perhaps in the US where just financially it's not affordable for people to keep taking in the long term.
I can't help feeling that in the future we are going to have to look beyond those 2 years, and I can see that we will be using things like lower doses and combined with other interventions like physical activity and diet, because you just don't want, I just don't think you want people on these drugs really long term and into old age, where you've got polypharmacy. We're going to, I can't see that we're all going to be injecting ourselves with these when we're sort of in our 80s and 90s. But I may be wrong.
I was speaking to a colleague in the US who's a CSO of Fractal Health, and they're developing gene therapy where essentially you inject twice an AAV into the liver, and then also into the pancreas, or it goes into the pancreas, and then you produce forever a GLP-1 receptor agonist. And his view is that in the UK, we have quite a sensible approach to these things, where in the US they don't want to take drugs and they want a lifelong obesity intervention to be able to do this. So that's one part of the future, and the other part of the future is, of course, our way of doing it, which would be to do this in a moderated way.
Brilliant. Last question, I'm afraid, just run out of time.
So it's a bit of a two-part question. One is, do we know if over the long term the use of these things wears down? In other words, efficacy goes down. And
Should we take that one first?
Okay.
If you look at the curves, yes, most of the curves that I showed, they go down and then they stabilize. And depending on which drug you're using, some of them stabilize faster, some of them stabilize at a different level, but most of them do seem to wear off. So, well, not wear off, just you have less and lesser effect with time.
And do we have any view on any long-term effects? Have we seen anything? I mean, the partial versus the full, but the partial's new, but the full's been around for 20-some years. Have we seen anything emerge that would cause concern for you guys?
I don't think so. I mean, the main side effect is nausea. There was this story that they might increase the risk of pancreatitis, but I think I agree with David, that's pretty much been debunked now. And all of these other presumed benefits or expected benefits on neurodegeneration, risk of cancer, obesity associated with cancers, and those sort of things strongly point towards these being overall net benefit. And you can probably look at bariatric surgery as also another way of understanding what the benefits of long-term weight loss are. I guess the reduction in lean muscle mass is is maybe the one which gives people the most concern at the moment. I don't know.
Vision loss. I'm a vision scientist. It's a lot of neoplasms going on, especially for people. Your question over 50, it's visual impact. People will be blind, taking two drugs.
Going blind.
Yes.
Is that in the context
It's
of
not
people with diabetic retinopathy?
Because human vision is very different from mice one, so this effect will not really studied.
Okay, that's interesting.
But it's really very serious too.
Thank you. We've run out of time, coffee seconds. So huge thank you to our panel for such clear presentations and for time as well. Thank you so much.